Thank you, Bengt, for a nice introduction, and I really am grateful for KBA and Molecular Frontiers and all the people who really worked hard for this, and I'm very much pleased I can come here to give a talk. Now, mirror image animals, mechanical manipulation and the point mutation, very strange, isn't it? We had such a beautiful talks before me, so it's very hard for me to try to keep the standard, but I should do my best. Now, on the right-hand side is the very famous painting by Leonardo da Vinci. On the left-hand side is the drawings by not famous at all people, me, drawing, and this is showing animals has bilateral symmetry. You know, this is the left-hand side, the right-hand side, the mirror images of each other in the middle. So this is showing. Although we are bilateral, but our body is asymmetric. That is chiral. You know, you can see the heart or stomach and the gut go to one direction. And this is determined by gene. Genetically, it is determined. If you go, exceptions, this flatfish, these snails, these are chiral. But again, these are determined by genes. And I had a project called chiromorphology. This is actually I made myself from the two words, chirality and morphology. I tried to work it out at the molecular level to the macroscopic level, try to look at the linkage through chirality. And I had two research groups. I'm a chemist. As Ben said, I started as a crystallographer, then I went into spectroscopy, but you find I'm also a molecular biologist, developmental biologist, perhaps I can say it now. The chirality is chemistry group. I'm working on chirality, recognition, generation, enhancement, and measurement in the solid state. This is chiroptical spectroscopy. I'm not going to talk about it. And another one is the biology. And I'm working on the origin of the chirality of snails. Now, this is the logarithmic scale in meters. So this is maybe 100 meters going down to 10 to minus 10, that is angstrom. And in the vertebrates, it's known these nodal and PTX2 genes, they are somehow involved in the left and right body plan, but it's in the vertebrate. So this is, you hear about two genes, please do remember. So what I'm doing is try to work out what is a gene which is determining our body chirality and our body plan. So that's what I've been working on. And I started working on snails, and people think, why on snails? And particularly, I'm working on snail called Limnaea stagnaris, because I think Limnaea may become one of the best model animals for studying the organismal chiromorphology. That's my belief. But I had a difficulty, because if you work on C. elegans, Drosophila, or Xenopus, I have many people to work with me. But snail, I'm really, the very few people work on that. So it's very difficult to find people to work, collaborate. So that I suffered a lot. But it's very interesting. Why? Both dextral and sinistral one, that's found in the wild. And I said, snail or flatfish is genetically determined, but normally you have only one side. Limnaea, you have dextral and sinistral in the wild. The dexterity, 98% is dextral. And another point, snail handedness depends on the direction of the spiral cleavage of embryos. That was determined in 1984. Now, I think I just missed one. Yes, this is the snail I've been working on, and this is the right-handed one, dominant one, and this is the sinister one, so this is only 2%, but this is in the wild. And so the direction of spiral cleavage, what does it mean? This is called spiralian. So it does the spiral cleavage at the first, third, or fourth, or maybe up to fifth cleavage. So it's a fertilized egg, Then it becomes the 2-cell stage, 4-cell stage, 8-cell stage. Then go with first, second, third cleavage. And if it goes to sinister one, third cleavage is very easy to see because from big 4-cell, the small 4-cell comes out. You go macromere and micromeres. Now, if it goes anti-clockwise way, it will become sinister. And if it goes the clockwise way, it becomes dextral one. That was reported 1894. Okay? So that means it determined the very early stage of embryogenesis. It's really nice. Then it's hermaphrodite. That means it can be either father or mother. That means you can do the self-cross, and also you can mate each other. That's not easy, but you can do that. And body asymmetry follows a maternal mode of inheritance. That was mentioned 1923 to 1930, still bent by Cott and Diver. But this is well before Watson, Crick, Wilkins, or Franklin, or Chargaff. All these people worked on DNA. That is 1953. So that is Mendelian gene. But they said 1920-30, it's determined by single gene. We worked, and we know it is single gene locus that we've done 2004, 2009. Then last year, we published a paper, we determined that single gene at last. So I can talk about it now. This is how it does. I'm not going to spend too much time on it, but pure dex, the large D, and sin, sinister, small d, you can cross it, then you get this. It depends on your maternal inheritance. That means even you have a sinister one, if your mother is right-handed, you are born with a body of right-handed one. And this, you can self-cross it, and this goes to 3-1. Now, this right, left, and this right, left. Depending on the mother's gene, your body is determined. What does it mean? It determines a very early stage of embryogenesis. So that is really wonderful. And worked a little bit, and to make it easier, people worked on L. peregula. I worked on L. stagnaris. Why? Oh, before that, I should a little bit story. Yes, in 1894, the Cramptons told about the spiral cleavage. 93, 1930s. So these people say it's a single gene, but it's a Mendelian gene. Then there's one paper, 1982, and it says, Freeman and Landuris, very famous one, a cytoplasmic factor present in the cytoplasm of the dexter egg of L. peregula has an activity that reverses the cleavage pattern on the sinister eggs. This paper appeared in the really professional developmental biology paper, Rue's archive. I was a pure chemist at that time, but this paper was just introduced in Nature, News and Views, one page. That stuck in my mind. But I realized this is not reproducible, and this may not be true. But this made me work on this work, and I started the project in 1999. Okay, then I decided not to work on L. peregula, but L. stagnaris, because this is much bigger. I was a physical chemist. How can I do developmental biology? And this is the egg, L. peregula's egg. This is egg cocoon, egg mass, maybe 20, 30 eggs in it. This is a stagnaris egg cocoon, maybe 50 or 80 eggs in it. So I decided, I'm not a biologist. Perhaps stagnaris, I can do it. And that was a very good choice, actually. And we started, I went to Holland to get these snails, and we started to how to make a big tank like this, and we really have to work hard because we cannot publish a paper. We are getting an expert to cleaning up the droppings of snails. No, I cannot write a paper on this, so we have to make it easiest way, automatic way to do, put in the water and cleaning it, and the UV lamp, etc., etc. So we've done very hard. And this is a life cycle. Now, this is the egg mass I showed you, and just a little one this is egg. Think, if it's a hen's egg, it's covered by white shell. it hasn't got shell. These are white, sort of albumin, and this is the egg. If you look at the egg, this is the one-cell stage, and it goes to blastoid stage, we call it the gastrous stage, trochophorus stage, Berger stage, juvenile stage. This is still inside this egg capsule. Then it hatches after 10 days. Then it becomes bigger, maybe after 3 months, 4 months, start produce next generation. So perhaps I can work on it. So this is the life cycle of L-star analysis. We have to establish all that. Then I decided to observe the development very carefully. So on the left-hand side, left-hand side is the dextral one, and I said it's rotated clockwise. We just classified clockwise, partial clockwise, neutral, partial anti-clockwise, anti-clockwise. Of course, there is nothing like this. All are clockwise rotation. And of course, some die because of some problems, deformed, but most of them becomes healthy dextral snails. If you look at this. This is, it should be anti-clockwise, but you'll see many, which even clockwise rotation. So this is what actually Freeman's paper must have done. They just fix the cell, just kill the cell with dye and observed only 8 cell stage. But you don't do anything, just observing it within a capsule, but it does all these strange things. And I know the reason why sinister one is not good and the one dexter one is so robust. There is a reason. And of course, it's determined by the gene. So then I try to repeat the Freeman's experiment. So we have to work hard and, just under the microscope, you hold the egg and you injected it or take things out. It's very, very difficult, but you can do it. Now, this is for the cytoplasmic transfer into the dextral one-cell embryos. If you put sinister cytoplasm into the one-cell stage, nothing happens. Okay, you have to take eggs and some from the same batch, same cocoon. You keep some for control. So this is the pair. So I've done many, and hardly any change. Then what's happened if you do it other way around? This is the cytoplasmic transfer experiments into the sinister one-cell embryos. Now, this experiment 1 and same cocoon control, you said, oh, you will see the clockwise one. This might have been observed by the Freeman, but if you look at it, control has that one. So you cannot jump into conclusion. Now then, I decided to put the sinister cytoplasm into the sinister one. Same thing happens. If you put Lucifer yellow, this is fluorescent one, just like a control. Same thing happens, even you can go to clockwise one. So you have to be very careful when you do the rotation, because sinister one has a reason to go either way and may die. So I observed things. The dominant dexter and the recessive sinister are not mirror images of each other at the crucial chirality determining step. The crucial chirality determining step is when it becomes from 4 to 8 cell stage. So this is sort of figures shown in most of the textbooks. Embryology or developmental biology, it says it's the sinister one, dexter one, sinister one, dexter one, 1 cell, 2 cell, 4 cell, 8 cells, 24 cell stage, 1, 2, 4. And they said it all goes to mirror image. Another textbook says same. But we realize if you look at carefully, it is not the case. And that is important for the handedness determination. So if you look at the top one, rotate this way, that's sinister one. If you look at this way, that becomes right. And the torque is amazing. I really want to work with someone. What is this? Actin is involved in this one. So really, someone said energy is important in actin, and it is. So this is 4 to 8 cell stage, and the sinister one, dexter one become like that. So, but if you look at it closely, this is from 4 to 8 cell third cleavage. For the dexter one, here already the cell, we call it macromere or blastomere, is deformed towards, protruding out towards helical way. And spindle is tilted to the helical way. And then when it comes out, it's already coming out to the helical way and it cleaved off. And it becomes dextrous. No exception. For the sinister one, at this stage, just cross, there's no chirality. And spindles, also no chirality, radially and going up this way. Then the next stage, the micromeres comes out just above. So again, there's no chirality here. Then it twisted to the right-handed way, and when it cleaves off to make 8-cell stage. So it can, something happened, it can go to the right-handed. So that is not robust. This is really robust because chirality is determined, destined, much earlier stage. So this may be the case. And we checked, oops, yeah. One is missing. Okay. Well, and this is the linear stagnized sinister and dexter. We can see SD, we call the spiral deformation, SI, spindle inclination, is displayed only by the dominant dexter one, and there's no SDSI. But if you look at sinister only one, Pfizer acuta, it's everywhere, actually. In the pond, if you go, you will see Pfizer acuta. It has SDSI, but it's in the mirror image way. So it's quite interesting. Now, then, I look, what is actually involved in the molecular way? This is the dexter one. If I do the inhibition by actin polymerization, there's no sort of membrane, and there's no SI either. So spindle is not pushing the cell, but actually cell is deformed and spindle tilted, following the shape of the cell. And if you do the inhibition of microtubule polymerization by nocodazole, for example, of course there's no spindle, but you can have a substantial spiral deformation. And of course here there's no SDSI, so you won't see any difference. So we can see actin is important for this handedness determination. Then we did a very old-fashioned classic way, that is like Mendel's peas, we just have to cross it. So pure DD and pure small d, if you cross it, left-handed, right-handed is very difficult, but you can do it. The F1, you have a 50% dexter, 50% sinister embryo, and because this all produces dexter 1, then you cross this with pure sinister one and make this mother. Then you have to wait for the next generation, what sort of gene it inherited from the parent, or father, or mother perhaps, this one. Then if it has this red dot, that is the handedness determining gene, it produces offspring, a right-handed one. If not, left-handed. So you have to pick up. You have to wait a little bit. Then you cross this with this pure one, and you keep doing it. It's not that simple, right? It's very difficult. But we managed to do it, and at the current biology paper, it was at this stage, 1.6% dexter and 98.4% sinister one. We kept this. Eventually, we got congenital, that means F10, 0.1% dexter, 99.9% sinister genome it has. And within that 0.1%, if you inherit the dexterity determining gene, it always shows SD, spiral deformation, spindle inclination, and it becomes dexter snail. If it hasn't got it, then no SD, no SI, and it will become sinister. There's no exception at all. So it must be in the single gene locus that we found. So it's just a famous snail appeared, and also it's in dispatch, and also Science wrote about it, so quite happy about it. Now, then I thought, if that third cleavage is so important, what happens if I push it mechanically to other way around? And of course, my technicians laughed at me. No, Prof, again, you come up with very strange idea all the time. I keep constantly, well, why can't I do this experiment? You get excited. And they're all much more calm and quiet. But anyway, I persuaded my technician. He's very good at it after half a year or so. So, what we did is like this. For the sinister one, it's easier. So I asked him to start from sinister one because chirality was not introduced earlier stage. This is the third cleavage. You pushed it by thin glass rod and put it here and wait until the cell contact is established. For this one, we have to start a little bit earlier because chirality is introduced earlier. So before this, just start to push it and wait. Then we culture that. That is again very difficult. Imagine you take the egg, hen's egg, take the yellow part, then you have to make it big to become adult. It's very hard, but I just make many nursery houses, nursery rooms, but eventually managed to do it. So this is how we did, and not easy to see it, but we just push it, push it, push it. And I'm not going into the detail. Now, this is the result. If it's the sinisterized dexter snails, so this is the trochophore stage, Berger stage, and the juvenile stage. So it's true. This is trochophore, Berger, and the juvenile stage, and these are adult. If the sinisterized dexter one, it's not dexter, but it just become sinister snail. And if it's a dexterized sinister snail, it's become just like sinister snail. And all the inside, we just open the shell and look at the inside. Everything internal body, its chirality is converted. Why? Oh, also, before we're going to say why, I just let you know, this normally, sinister snail becomes juvenile snail, adult snail, then it becomes sinister, and it produces sinister offspring. What happened to one? I just pushed it mechanically. Now, it becomes dexterized juvenile, dexterized sinister adult snail, but I haven't done anything to the gene. So it goes back to the chirality. It's dictated by the gene. So it's really fascinating, isn't it? So it goes back to the original chirality. So it hasn't done anything to the DNA, but we can change the chirality. So, and as I told you earlier, nodal PTX, that is for the vertebrate. It has been known for 7.5 days embryo. The cilia is formed and is tilted one direction, it's rotate. It produces some flow to one direction. And at the end, that will become left-handed. And 8.5 days, so the nodal appears on the left-hand side, and the PTX, that determines the body-handedness. And it has been known for others, if they change the direction of flow, this is a very nice work by Nonaka, the Japanese guy. It will change the chirality, they say. And so mouse, chicks, xenophus, zebrafish, but not known for the invertebrates, or some invertebrates, not for this class. So we did the cloning, and we found nodal is there, and if it's the sinister one or dexter one, it appears in the mirror image site, and at a very early stage. You know, we had to put two together to take a one-shot photograph. Otherwise, they may say, you can cheat it to put it the other way around, reverse it. So we had put the dexter and the sinister at the same stage, developmental stage, and took the nodal, and 33 already, 49 to 64, PTX, around the 64 cell stage, already there. And they're mirror image of each other, and they stayed until to sort of gastro state. So this is the nodal gene, and left and right, and this is the PTX. So what's happened if we invert it mechanically? So that is what it is. This is a 49 cell stage. this is reverse dextro embryos. So it should be dextro one, but the place exactly similar to the sinistro one. So if you push it at the very early stage of embryogenesis, you change the expression site at 49 cell stage, then after that, everything goes the mirror image way. So this is much later stage, and this is Turcofer stage. You can see, again, it's coming to mirror image. So that is the reason you had a very healthy mirror image creatures. So it just summarizes it a bit like this. If I do the pushing earlier from 2 to 4 stage, it go back to the original. So it's important to do this manipulation at the 3rd cleavage stage, 4 cell to 8 cell stage. Then you had mirror image expression site of nodal and PTX gene, and you had this healthy adult of mirror image. Then the offspring goes back to the chirality dictated by the gene. So it's really fascinating. So this is sort of a summary. Oh, the clock is not going, but I hope I have more time. Then, this is appeared in Nature and this. Then I thought, Limnaeus stagnaris, I said, it's dimorphism. It's very rare species. You have dexter and sinister, both in the wild. So I thought that may be the reason I can do the trick. I can change it, swap the chirality. What happened if I do this thing to the Phi circuitor? There's only sinister one only in the world. If I can do the same thing, there must be a common mechanism for handedness determination. And of course, I succeeded it. So I could create the never existed right-handed Phi circuitor. And that offspring goes to become the sinister one, back to sinister, just like this one. So this means there's, and you can see it, no exception. That means there is a common mechanism for the handedness determination in the sea snails. So then, of course, I've been working on the identification of the handedness determining gene. People looking for 1894, people wondering what is the gene. I've been working on it and had a terrible time, and it appears. And also, I've done the CRISPR-Cas, but unfortunately, I cannot talk about it today. So some other time, next time. And this one slide contains maybe 13 or 14 years, our hard work, joy, tears, and sweat, everything in it. So it's really terrible, but I just want to say it. Single-handedly determined gene locus was mapped by genetic linkage analysis, back cloning. It's terrible to make a back clone library, but we did it. Because no one wants to work with us, because I'm working on snail, not the C. elegans or Drosophila. And chromosomal walking using pure large D, and small dd strains and F2, F10. This is a congenic one I made, back cross lines. And then shotgun sequence of the 7 back clones were assembled into contigs and placed on the physical map by alignment with AFLP linkage markers. Then quantitative real-time PCR analysis. We did it, and we thought in the blood search, and we thought about 15. Then we checked the maternal one, because I know this, it has to be maternal one. So 13 maternal gene we identified. Then out of that 13, it has to be something to do with actin related one. So picked up 5 actin related one. Okay? Then full-length cDNA for both dexter and the sinistro strains was cloned by RT-PCR. Then we did 5' and 3' race, and sequences compared. Oh, this is 15 years of work, all right? So this is, if you know it, this is the part which there's no recombination. And I hoped it's much smaller area, but unfortunately, it was this 780 kilobase locus. And just a little bit, but anyway, these are backgrounds, 7 backgrounds overlapped. And out of that, these are the 15 genes, and red one is a maternal one. From that, I checked the actin one, so that is the fat, this is milk and fry, and DR1 and DR2. These 5 genes are related to actin. So we've done all full sequencing of this. And this DR1, DR2, this is a tandemly repeated one. The DR1, so this is one. This has homozygous point mutation. Here, DEX, this is a C here, CC, AACCTC, and here AACDCA. So one C is missing. This is the reason it becomes left-handed. Okay, so it's amazing. So it looks like this. This is the Dexter gene. It has DR1, diaphanous is DR1 and DR2. The sinister one is truncated very early due to frame shift mutation. And DR2 is exactly the same. It has many functions. I cannot talk that much about it. So it does everything. But DR1 and DR2 have 89.4% homology. So even it loses it, it survives. So that's the beauty thing. If you have a dimorphism, you can work on this because it has a tandemly repeated gene. So it is doing very important work. So that's how we did. Then we tried to work it out with 89.4% homology. How to discriminate DR1, DR2 is very hard, but we managed to do it. So this is the quantitative PCR, and this is for the LSDR1. You see, the one cell stage, the Dexter one, has a lot of this LSDR1, this is the messenger RNA, and it drops sharply, but for the sinister one, nothing. And LSDR2 both has some, and it drops. So this is the case. Then we did WISH for whole mount in situ hybridization. And we do it more carefully. Here, this is the one cell stage before first polar body extrusion. This is the one cell stage after second polar body extrusion. Second, 2 cell stage, 4 cell stage, 8 cell stage, 16, and 24 cell stage. And DR1, for the Dexter one, yes, you had some, but it's going down. But here only first polar body exclusion you have, but nothing. That means it's already translated in the oocyte, and it has it as a protein, and that is working. LSDR2, it has to certain stage, up to 2 or up to maybe 4, some is 16. In the 16 cell stage, where at last zygotic gene is getting to expressed. So up to 16, the present from the mother, you are working on it. So that is a beautiful case because system is very simple. And you have to see, there is no localization, okay? But I had a bit of problem. Some incorrect paper appeared, and it was really embarrassing. Like showing this, and they cannot discriminate DR1 and DR2, and they show these sort of things. These are the cause of the chirality determination. It's totally untrue, and not only that, several other results are not correct. So it was very hard for me. But anyway, that is it. There's no localization. And we did the Western blotting. So I thought if I can do the monoclonal antibody easily, listening to Leroy's talk today, it's easier. But anyway, we have to work hard to discriminate this DR1 and DR2 protein, and managed to do that. So for the sinister one, we don't have any. But for the Dexter one, yes. And it stays until blastostrage. This is amazing. And for this chondritic one, 99.9% sinister, 0.1% the Dexter one, and if you inherit the Handes determined gene, you have this, but if you don't, there isn't. So it's very clear-cut. So we're very, very happy about the results. But if the century-old gene is determined, more questions arise. Now, we've done this. This is the linear stagnus, peregrina, and the Dexter one, you had both type 1, type 2. And so if it's missing, type 1 is missing, then you may think, well, DL1 may be responsible for a right-handed one, or the DL2 is responsible for SDSI. But then you work on Phi circuita, left-handed only, or indications extras. It has only type 2. But it does SDSI, and it does other way around. So what is determining the direction of rotation? We start thinking about it. And there are 2 tandemly repeated ones. So originally, all snails may be left-handed. When they made a copy, changed, maybe transferred some role to the other gene, we don't know. But it's very interesting. So we start working on it. But again, it's very hard. If you do some work on this one, it all dies because there's only one copy. But this one, you have 2 copies, so we had advantage. You can work on it. So I'm doing this. And there are various, okay, soon I have to stop it. And this sort of different species have a different way. But one thing I think is quite nice is, for the vertebrates, human has this. There are some people who has complete mirror image, situs invertus, and nodal gene expressed 8.25 days, and the cilia started to appear 7.5 days. But for my case, after 3 hours at the cell stage, left and right hand determined, and here is the zygotic gene started to be expressed, and this is the organizer cell determined. So, and this is the nodal. So it is a really simple case I can work on. And using the snail, I can go into human beings work. And this is the main co-workers. They are the students or technicians. And for the future work, I'm not going to do that much about it, but really want to connect this to the amino acid, D-deoxyribose, D-ribose, homochirality of our biological world to join to this 100 determining gene. And also, I want to find out when the differentiation starts. So I started to work on, because D is something too special to go into this, eventually go to nodal gene expression, we know that. But when does it start? When does the differentiation start? Started working on a Raman spectroscopy and also mass spectroscopy. But of course, I don't have many students to work with me, and really want to work with many people. So this is really I want to do, but more fascinating story. So this is the last slide. And this is dear, so I'm very pleased to hear. But what it actually said is, give me a cup of coffee, and after my talk, I'm here, you want to say this. So this is the end of my talk. Thank you for your attention.